All right. The diehards have stuck it out. I think folks that maybe imbibed a little, little heavily last night are slow to make their way to a Sunday workshop. So thanks for joining. Let's see. Oh yes, our slides are up. Fantastic. So this morning, this is workshop number 4, partnering with BioFoundries. I'm James Gardner. I'm a program manager at the Agile BioFoundry, as the slide indicates. And so what we will have today is a slightly different format than what we had yesterday. It's a more of a traditional panel where we will have the panelists offer up a 15 to 20 minute presentation of their work and the ways in which they partner either with outside groups or as an outside collaborator, how they have partnered with BioFoundries. So that will include folks from the academic sector as well as BioFoundries from across the globe. And I want to kick it off a little bit by giving you some insights into how the Agile BioFoundry at the national laboratories in the United States conducts its BioFoundry collaborations. Okay, so just quick AV check. Are we good to go for the online Zoom? All right, fantastic. You'll also take note that I've put my email address up there. So if there are questions that arise that we don't get to in time, by all means, please do email me and we will enter those questions into the record and get back to you to the extent that we can with answers to your questions so that we can have a more kind of robust value add to this workshop. All right, so again, we will have a series of presenters, and that will include Marco Storch. I hope I'm not butchering your name, Marco. Allison Rice, Mark Blenner, and Yinji Tang. We'll take a quick break and then reassemble here for that panel discussion. You know, I want this to be something where you feel you are coming away with some actionable information, a true to form workshop. So, I just want to give a little bit of intro to the speakers. That's Marco Storch, head of the synthetic biology and automation at the London BioFoundry. Allison Rice, business development manager at CSIRO. Mark Blenner, who is an associate professor at University of Delaware, and online is Yinji Tang, professor at Washington University in St. Louis. I'll give a more proper introduction to each of them just prior to the panel discussion, but they will also have an opportunity to introduce themselves by way of their work for each of their presentations. Each of them have really contributed in a very significant way to the body of literature, as well as to BioFoundries and the way that BioFoundries have developed for the GBA and beyond. The Agile BioFoundry, and I'll keep this very brief, but the Agile BioFoundry is a consortium of 7 national laboratories or the programs therein that make up the kind of flagship activities in synthetic biology for the federally funded research and development centers within the United States. And so these are researchers who have a long and storied careers in their own right and have come together over the past 7 years to sort of synergize the work that they do and perform various experiments and various lines of research for collaborators, as well as for core research that feeds into the capabilities of the Agile BioFoundry. The hexagons here kind of represent the why, the how, and the who of the Agile BioFoundry. The why is really focused on the Bioenergy Technology Office's core mission of industry decarbonization and the production of sustainable aviation fuels and other types of fuels that are difficult to electrify, essentially. And so the work that we do really centers on how to turn difficult products that would otherwise come from a fossil feedstock into products that would have a more sustainable horizon. We do that, of course, through the DBTL, through developing the appropriate infrastructure, and through the work that all of that comprises, including the difficulties associated with the onboarding of non-canonical hosts and a whole host of capabilities that make up the DBTL cycle. With regard to the who, it sort of is, at least from the perspective of this workshop and from the perspective of what the Agile Biofoundry is capable of offering the community, it's really sort of twofold. One is indirect, and that is through workforce development, through the training and education that the staff gets directly, as well as the kinds of workforce development opportunities that sort of take place tangentially through the various locations of the Agile Biofoundry spread out across these 7 national laboratories. Kind of more directly, it really involves sort of the crux of today's discussion, which is partner development. And so kind of in keeping with that, over the past 7 years, we've managed to develop partnerships with 26 groups. And so we have 26 projects essentially that are either completed, that are research that is ongoing, or research that is kind of in the contracting phase. That really spans the gamut from companies that are strictly focused on a specific product or product line to companies that are more specifically focused on tool building. Agilent is a good example of a project that completed last year, where work was developed to develop various kinds of omics workflows. So not a product in the strictest sense of the SynBio definition, but certainly an important value add for the community. And that's just one of numerous examples. Okay. Now, with regard to partnership development, you know, it's an ongoing thing for us in terms of understanding how best to do it. We are blessed by having a $20 million annual budget, and currently about $5 million of that is devoted to the work that we do in conjunction with our partners. And so we offer a variety of funding opportunities that include sort of the traditional open funding opportunity that is the most attractive typically for working with industry, where they have the direct path to offering up the required cost share. Other types of sort of collaborative funding opportunities that we have developed or are currently developing include one with the National Science Foundation we kicked off this year, along with a minority serving institutions funding opportunity we also kicked off this year. And we're pretty excited about both of those new developments. We are in conversations with BioMade and looking forward to seeing how a collaborative funding opportunity with that group might evolve. And I'm not altogether sure if everybody is familiar with BioMade. I certainly encourage you to explore that organization, really focused more on the higher level of technology readiness, looking for something closer to commercial deployment, and funded partially, at least, through the Department of Defense. And then there's sort of the so-called funds in opportunities, where we are looking to interact with groups that are all about leveraging either VC funding that they may have already acquired, or serving as, say, subrecipients on grants, as well as sort of more direct type funding opportunities. So there we are kind of in these early stages of development, and we're excited to kind of continue that path. What we've discovered is that you learn a lot along the way, and part of that continuous improvement is in the realm of standards. We talked a lot about standardization yesterday in an entirely different context, but the analogy holds, and it's important to think of this type of or this side of the work as something that can be standardized in its own right. And so we're looking for ways to do that, to speed up contracting, to better assess our impact kind of retrospectively, as well as get more IP licensing out the door. So there are any number of things that we are doing to continuously improve. I'll leave it at that, and I'm happy to take questions, but I really want to make sure that we're giving the lion's share of the time to the speakers. And so for that, I'm going to now switch over to Marco's talk, and maybe kind of just doing this a little bit on the fly. I think what I'd like to do is, Marco, offer up your description. So I think what I'll do to do that justice is jump down and go to your talk, bring that up. And all right, so if you could switch over to the screen, and I will launch. Okay, and one moment. Okay. So Marco Storch joined the Imperial College in London in 2012 and worked as a research fellow on automated DNA assembly and directed evolution projects. And in 2019, he then joined the BioFoundry leadership team, where he focuses on the team, the platform, and business development. And without further ado, Marco. Thank you very much.
Good morning, everybody. Thanks very much for setting up the session so nicely, James. Yeah, hi, I'm Marco. I'm running the BioFoundry in London together with Paul, that you know very well. And we also have Kaltum and Chris with us, who both have a poster that you might have already enjoyed yesterday. And then also Kiva will give a talk later today. Yeah, and then Kiva will also give a talk later today on her work on cell-free. Also, thanks very much for the organization of the event. Fantastic week we had, and we had the chance to have a bit of fresh air start of the week as well. So that was much appreciated. So it's more like a working document, not so much kind of a presentation in the spirit of a workshop, really, right? And so I think I want to orient you towards like what kind of organization are we, and then how do we go about developing like our partnerships. And so just very brief, so we have around like 10 people as a core staff. Then we have people that come to work with us. These are postdocs from groups we work together with, or from industry partners. The BioFoundry was built with around like 6 million CapEx. And so at the moment we run it on a budget of 1.5 million roughly. There's a plus, and we work really hard on this plus. So trying to kind of, be more ambitious on things as well. We have a whole range of projects, both academic and industry related projects. And we managed to kind of get to 7 to 10 publications a year as well, right? So kind of staying in the game there as well. Interestingly, per year, we have like one startup that's created from members of the BioFoundry, right? So that kind of indicates already kind of an affinity to entrepreneurship that we're very proud of fostering within the team. And in terms of like real estate, so we have like 250 square meter lab space. It's quite packed with lots of cool kit. And if you like to learn more about this, feel free to have a look at the website. What's really good for us is that we like located in the iHub. So that's a purpose building for startups. So we kind of the only pure academic group in there. All the other groups are actually industry partners. And then if you look at like our wider context, focusing on London. So we are based in West London at the new campus that Imperial College built there. And so in the iHub building, the innovation building, we have a couple of startups we're working with on the left-hand side. But what's also really interesting for us is that like now in London, a lot of new kind of companies grow and also base themselves in London. So you can see that they're basically kind of almost creating a circle around the city itself. And so we really enjoy working with all of them and have small and bigger projects across. And I think that's also a really interesting development in a sense that on the one hand side, institutions like Imperial College really invest in these campus models where the co-location between academics and industry is made very easily. Of course, London offers like 3 fantastic universities plus Oxford and Cambridge, so have a lot of talent that's ready to join the workforce on this. We have hospitals, so fantastic access to patient samples for this type of research. And of course, you will know that DeepMind, Google, Microsoft have like massive bases there and really commitment to the city, right? So it's really kind of shaping up to become a very interesting, exciting kind of environment. And so I think one of the few silver linings of the COVID crisis really is that a lot of office spaces are empty in the city now, and the real estate developers are super keen to change this into lab spaces and offer it out to cheap, right? So that's really a fundamental change. Like London was never affordable for startups to establish themselves and grow, and that's completely different now. And I think in my mind, that was kind of the missing puzzle piece for London to maybe really shape up to this biotech hub that we would find so exciting. I guess like another kind of example for that is that real estate developers decided like to build the biggest, the largest European dedicated lab building in London as well, right? So it's kind of another vote of confidence for this scenario. And of course, London being the financial center, you have a lot of capital and kind of a growing VC community as well, another important ingredient. So now I want to go through a couple of different partnerships, starting with academics and then go into industry. So on the academic side, really, that's kind of our bread and butter and our comfort zone, right? So we grew out of like a very enthusiastic synthetic biology community. And so we define a couple of topics that we're really keen on developing and spending time on. And then kind of we align this with the key partners. The most important one maybe being the Imperial College Center for Synthetic Biology. So we work with a lot of colleagues over there, and of course the proximity is really useful for that. But we also worked a lot with the NHS as part of the COVID response. So we set up like the COVID testing in this context where 1.5 million tests were performed in the end. So that was a good example there. Of course, like the Global Biofoundry Alliance, the National Physical Laboratory, and the Dementia Institute as well. And so I don't want to have so much because like that's kind of a setup that we understand quite well. But then if we have a closer look at like how these activities actually shape up and what we can maybe learn in terms of strategy for biofoundry development. So I think we've seen again and again, and that's only proving that the biofoundry is acknowledged as a fantastic partner for any grant. So we get a lot of interest from different departments now, from chemistry to medicine and so on, to actually work together with us and leverage technology and know-how that we can offer. And so that helps really with securing grants, of course, and our postdoc runway. Then you might have seen in journals like Nature and Science, the postdoc crisis is a thing, certainly in the US. I'm not quite sure how much it's the case in other countries yet, but maybe that's coming as well. And I think we really like spoke also like to academics who really worry about how can you make like a postdoc position as attractive as possible to attract talent, right? And that's again like something where the biofoundry angle works really well. So that's for us again like an interesting kind of strategy to pursue. It's of course also a place for education and for workforce training, right? So people that come to work with us or our core member team, they really educate themselves in automation and a lot of like the cutting edge technology and having access and exposure to like certain systems that you wouldn't in a normal lab, right? So that's fantastic career development for them. And as I mentioned in the beginning, we're also quite keen on entrepreneurship as an activity. So there's courses and so on. But what's really exciting, I think, is that people that work in our biofoundry have exposure to a lot of startup stories. So you talk a lot with them. There's a lot of kind of osmosis of information going on there, right? So that's real life experience rather than kind of pure courses. The drawback maybe is that obviously like funding cycles and so on, that's always unpredictable, right? So we cannot solely base our kind of financial framework on the academic side. And so if we then move on to like the startup scenario, so we're working with a lot of them, and as indicated on the first map, so we have like over 20 startups we're working with in different intensities. And so what you typically can do is obviously advise and give access to equipment. We can do very small work packages, for example, an NGS kind of project, sequencing antibody libraries with PacBio or similar. And then we're also kind of starting to offer something like, we think about like as a biofoundry residency, right? So the idea there being people get access to the biofoundry and you're not so much like looking after how many hours of plate reader were used, but people literally get a bench and get the access and also get integrated in the team and can learn from the team that we have in-house. So I think that's something that I'm quite excited about kind of pursuing further. And I think we have like early science that works really well for certain startups. Now, in terms of what we do, for instance, we have like 3 startups here. Modest Media is a company that works on cell culture media optimization. Syndo is a company that works on enzyme development for plastic degradation and pure affinity, purify deals with wastewater, specifically like PFAS molecules. And so like just based on the technology stacks we developed and expertise we have, that's quite useful. Those companies that have access to technology they couldn't afford otherwise, and also wouldn't have training for using actually. So there's a couple of very good examples there. And if I just like quickly try to summarize what we kind of learned so far, the projects with startups in terms of supporting a biofoundry financially, as you would expect, are probably not super helpful. But we feel it's a very important activity for a lot of other reasons as well. So typically, like you have to be very flexible. The costs can't be very high. You need a lot of attention. You have to develop these relationships with a lot of energy, which is also fun. And obviously, like you transfer a lot of knowledge, and so there's value in that. But again, it's quite hard to create a model where you capture this actually, like, and it's probably also not the essential bit of the activity in this case. So for us, it's really the real impact, like if startups can kind of thrive in the environment that we can be part of and support. So that's actually our success, right? So that's what we're going for here. And we completely accept that we won't make much surplus on these activities, but we feel there's a lot of other benefits to the ecosystem and also to our own work. And as I mentioned before, so we try to make it more flexible in the way that we just kind of sign up startups to residency models so that we have like less kind of overhead and kind of complications with managing these relationships. And I think what it really, how it also serves us really is that obviously it gives you exposure to a lot of different domains. So you can build up domain expertise in the things that these startups focus on. It also gives you credibility, right? So all these communities talk a lot to each other, right? So if they're happy with how it works, in their case with the biofoundry, that will bring on new customers, and hopefully all of these startups grow up to become SMEs, right? And then it's becoming also a financially interesting partner for work with us. And so at this point, then I want to move over to the SME example. So there's one company based in London called Procarium, and they really focus on the living therapeutics space. So they have a Salmonella strain that developed further, and they kind of leverage the ability of Salmonella to like home into tumor microenvironments. And so the idea here is like that you make the cell even smarter to then kind of, turn this into a proper cancer treatment. And this project is fantastic for us because it matches so nicely with all the pipelines that we've built in-house, right? So anything starting from high throughput strain engineering to all the analytics, the NGS, genetic stability, and all these other questions that we can answer really well based on the kind of workflows that we established. And maybe just looking at like the practicalities of like how such a more ambitious project actually came about and works for us. So as you would expect, like the initial negotiation was a bit prolonged, and so it took like 3 months to come to an agreement for like a pilot phase. And that's, I think, also quite important that these more ambitious projects have to be like milestone, of course, and has to be kind of a phase where you really build a trust initially, so that people might then commit to like more ambitious projects down the line. And in a sense, like that's a really good blueprint for us here. So after initial 6 months project, where a scientist from Procarium like was based in biofoundry, was very successful in pursuing this project, management was then like very easily convinced to kind of put more effort into this. And now we have a much larger team that consists both of like biofoundry members that are dedicated to this project, but also like a couple of people that are seconded from this company that are probably embedded in the biofoundry, can leverage all the kit and knowledge we have. And so things that we learned from this example is really like that it's all about trust at this stage, right? So you really have to get the critical mass together, and it also has to be quite a good product market fit essentially for this company, right? That the kind of abilities that you can offer and where you can compete with CROs, or actually like outcompete CROs essentially is the real aim here, that you're like in a good position. And so also, I think like the communication is quite important. So it takes a lot of my time actually like to make this project work, but it's well worth it. And there's obviously a lot of people involved. And also if you have people seconded from a company, obviously you didn't select them, right? So you have to hope for the best. But again, I think what we saw is that because the company could recruit based on biofoundry collaboration, that really helped them attract top talent. So it's another example of where the biofoundry brand is fantastic in kind of team development. And so I think also for us, like it just means we constantly have to look ahead to see like what people might actually need, because when you start this conversation, you want to be able to like execute within a month basically and kind of be on the road. And so that worked really well in this example. And so that's also something that keeps us busy, like trying to understand what is the next critical mass we have to build up to be able to offer something that's really attractive to SME companies that then can help us with developing our science, but also helping with the income that we like to generate so we can grow the biofoundry further. And so with that, then maybe I want to kind of switch over to the final topic, which is partnerships with manufacturers. We found it's quite important as well for us. And so we have kind of 4 really quick examples for that. The reason we're doing this is because we're very excited about what's next. And so a lot of these things are driven by capabilities that the next generation of machines provide you. And the first example maybe being here, DNA Script, with whom we had like a fantastic collaboration running for 2 years now. And they're also presenting, so you can learn more about the technology outside. And so we kind of been part of their beta program. So we got an instrument and tested it in a couple of different applications. And so it worked really well. It was also quite timely. It was part of the COVID crisis where you couldn't get hold of any kind of oligos that are specific to the COVID sequences. And so actually then we managed like with this machine to print both sequencing primers, but also Taqman probes in-house overnight, basically, right? So that was quite a game changer, what this machine enabled us to do. And of course, the long-term vision for us here is kind of completing the in-house SynBio stack, right? Where we can start creating sequences in-house and building it all in-house from start to end, having already all the analytics in place, right? So that's an ambition for us. And so we hope that like next year we can see activity in the space where we start building like 1 kb genes in-house as a collaboration, right? And again, it's something that we couldn't do ourselves, right? It's just like where we're fortunate enough that we have a really good relationship established with DNA Script now. Now a quick example is a company from Cambridge. So that's Nuclera. They're really focused on being able to manipulate like these little droplets that you can see in the video down there. And so you have the chance to like organize experiments and reactions at really small volumes in high precision. And so the first applications for them is like to just screen a lot of configurations for kind of expression cassettes versus lyset types to optimize this combination. And that's something that we ran through. And again, that works really well in our lab. Then something for next year, quite ambitious. So we had to look at the biofoundry here, right? And there's real ambition on the automation of cell culture. So now here's a company that basically aims to provide you like these 4 big towers that are meant to manage 200 plates. And so we will then see like start of next year how that's going in an alpha phase, right? So it's very early, but again, like part of horizon scanning. And then related to that, like we also work with Nikon on kind of the next generation high throughput, high content imaging systems, right? So that's all kind of linked up between the capability of creating the experiments in the first place, but then also like having an analysis capability that matches the throughput. So for us, in the summary on like the manufacturing side of things, is really like that it's a fantastic exercise for horizon scanning to understand where the field is moving, what's working, what might not work. I think in practice, it always takes a bit of effort to get these contracts in place. And so Imperial College is kind of helpful with that. So that's really good for us. And of course, also it means from our team, we spend time on it, right? So we want to be able to contribute data and app notes that should come out of these collaborations. And then finally, I think what's really important is that for manufacturers, actually, like again, the biofoundry concept is the best context to test these machines, right? Because often they have to work in a context. So have an upstream or downstream process that you need to actually say if this machine works and is helpful, right? And so I think that's another kind of unique selling point for biofoundries in this context. And so with this, I want to close. And I think I just want to like the argument that like these different kind of activities across academic work, industry work, but also manufacturing, all of those together have really the opportunity to like give you real synergy in these things that you couldn't achieve otherwise. And specifically for like an organization like ours. So we have to be very lean. We don't have as much money as we wanted to, of course. And so that's kind of a key element for us, like to try to combine those and leverage those different elements together. Okay, that's for me.
Thank you, Marco. So I think what we can do is hold questions unless there's some burning question that you want to ask Marco, but I think just in the interest of time, it'd probably be better if we stay on task and give the time to the next speaker. Allison Rice has a long history of leveraging her experience in research, education, and government to make connections, develop strategies, and manage complex projects for CSIRO's Brisbane location in Queensland, Australia. And Alison very recently moved on from her deputy director role of the synthetic biology future science platform at CSIRO to now is the full-time business development manager. So please welcome Alison Rice.
Good morning. Thanks, James, for the invitation. I need my.
Guess that would help.
That would help.
Sorry. Okay.
Thank you. So a bit about the synthetic CSIRO. CSIRO is Australia's national science agency and is devoted to developing scientific and technological solutions to Australia's greatest challenges. In 2016, CSIRO identified that synthetic biology was a key growth area for and an opportunity for Australia, but recognised that there was a very disparate synthetic biology community within Australia, and so with funding of $13 million, set about developing a program of Horizon 3 work, which in the space of industrial biotechnology, ag and food, health and medicine, environment and biocontrol, synbio-based tools, and also looking at the social, legal, ethical, regulatory, and institutional barriers to uptake and adoption of synthetic biology products. So the mission of the Synthetic Biology Future Science Platform was to build capability, community, infrastructure, and understand the barriers to uptake and adoption for synthetic biology and a bio-based economy. So the BioFoundry came out of the Synthetic Biology Future Science Platform, and it was established in 2018, initially in collaboration with the University of Queensland, and then in 2020 moved across to be wholly owned by CSIRO. So I want to talk to you a little bit about the Synbio BioFoundry, which is based at Dutton Park in Brisbane, sunny Queensland. So the BioFoundry is capable of DNA, is a high throughput DNA organism and engineering service, and offers design, build, and learn test facilities, test work for the academic and industry partners. And what's interesting is that we can actually work in quite a modular phase. So if people need to come with, they don't need the full suite of gamut of work that we can do. They can actually come in and just have design, build, or test services. And we can also refer on to other facilities within CSIRO in terms of metabolomics, proteomics, scale up, and fermentation. And we can also do a lot of strain engineering. And the team works with Saccharomyces, various strains of Saccharomyces, Pichia, and E. coli, and is also able to take on novel organisms. As I said, the BioFoundry was established in 2020. It took about $5 million over a couple of years to establish all the equipment, and currently runs with a dedicated team of 5 with Janet Reed, who you'll know as the facility manager. But we also have a new director, Colin Scott, myself as business development, but I'm not full-time on this thing. We have had a comms contractor, and then we have 2 technicians, Lisa and Candice, a project officer, Lee, and a data engineer. So they're the 5 people at the bottom of the core staff for the Synbio. And I'd also like to acknowledge the significant contribution of past team members, particularly Claudia Vickers in her role as Synbio, the director of the Synbio FSP, Marche Halolko, who was involved in the Synbio stack development, and technician Hugh Heyman Zump and Sam Wilson, and some initial BD work that was done by David Mitchell. So stakeholder engagement, we're a lot younger than some of the other BioFoundries you've been hearing about. And so we have a slightly different approach to stakeholder engagement. And one of the things that people like to see is machines that go ping, and everybody's really enjoyed the the tours that we've had here at Concordia. And so we've, Janet and her team have taken about 100 tours. We decided to keep track of them. And they range from school kids, which addresses the STEM workforce pipeline, to academic researchers, industry partners, state and federal government people, and foreign dignitaries. And everybody's really impressed. They come in and they don't know what synthetic biology is, and they leave having visited this really cool lab with a really enthused and excited about the opportunities that SynBio can afford and what the BioFoundry can actually, the role that BioFoundry can actually play. So the team also is active in scientific engagements. They speak at scientific conferences, they speak about the work that they do, but also about the BioFoundry. You've invited seminars. They've done an industry webinar for Twist, which was a global webinar, which was a really cool thing to do. And that actually brought in quite a few people, inquiries about, you know, could they come and work? And so it was a really great way to reach people who may not have known about us. We also do a lot of work within CSIRO. So CSIRO is like an organisation of 5,500 people across Australia, and not everybody gets SynBio. So we spend a lot of time talking about that. We've also got really strong internal engagement. And so CSIRO is always looking to the future, and the chief scientist, one of her missions is the labs of the future. So we're active, Janet's actively involved in discussions about what does the lab of the future look like? What are you going to need? The robotics, Internet of Things, the sensing community of practice, lab automation group. There's a cross-cutting capability for synthetic biology within the organisation, equally in engineering, cross-cutting capability, genomics. And Janet and the team have played a really instrumental role in the Institutional Biosafety Committee, which is really important because it gets more people on board understanding and streamlines the sort of the work that we need to do. In terms of external engagement, working with the commercialisation team, we pitched an idea to the University of Queensland MBA entrepreneurship program, whereby and the idea was that they would come up with an idea and they'd use the BioFoundry. So I got the job of speaking to this cohort of MBA students, told them about what synthetic biology was, what it could do, and why BioFoundry was integral to this process. And they came up with 6 ideas, 2 of them, 2 potential projects and 1 viable candidate. And so the viable candidate, the idea worked with CSIRO scientists, and they were successful in getting funding from CSIRO for an entrepreneur in residence. And then this young woman has now done further accelerator programs and has managed to attract $3 million in seed funding within 12 months, and expects to raise further seed VC. And this is all in the nutritional protein space. So it was a really wild idea that worked, and it was a really great way to engage a whole lot of people who'd never heard of synthetic biology before. The other thing as part of the Synthetic Biology Future Science platform was we recognised that the IGEM is a fabulous competition, but it doesn't suit the academic calendar of Australia and the Southern Hemisphere. And so with help, or that actually largely driven by fellows within the Synthetic Biology Future Science platform, we created the Oz SynBio Challenge, which is largely built off the IGM program. And to date, we've had 170 students run through the program through 11 universities across Australia, and they've been supervised by more than 100 mentors. And what's been really exciting about this is that these students have come away from this program and have become committed to going forward to do an honours or a master's program, both here in Australia and in Europe. And one's actually working in a SynBio startup, and the others are pursuing sort of entrepreneurial activity. So it was, again, an easy way to spread the word, which was, and get people really excited. And this program is ongoing. I really liked Marco's presentation about engaging with industry directly. We have an opportunity in Brisbane. There's a very large underground railway system being built, and one of the stations happens to be outside the building where we work, and they need to stick a building on the back of the station box. So we said, yes, you need a bioincubator. And so we've been actively working with the Queensland government, University of Queensland, the health service just next door, and the other, to work out what we actually need to develop a startup hub. And the biofoundry is one of the key pieces of infrastructure. So the long-term vision is to move the biofoundry from the building that it's in into this new mythical building. So hopefully when they again renegotiate, they change government, so we need to go back and renegotiate. But it's really key. We see the biofoundry as key, as a key enabling piece of infrastructure to bring together industry, to build that sense of the ecosystem, have spaces for VC. We're working actively with the Queensland government on industry attraction to get startup synbio companies from the US to come across to Australia. And by thinking about all these things now, we really hope that we'll be in a place like you are in a couple of years. Funding, the biofoundry was set up, cost about $5 million to set up. We have an annual running budget of about $1 million, but we have no subsidies. And so we need to work out how we're going to become financially sustainable. And so one of the mechanisms David Mitchell, who did some early work on the business model development, was he worked out that you needed to actually get some internal champions, people who would say, I can do this piece of work for you, my industry partner, but we really need to use the biofoundry. And we're starting to see that happen. We've also worked closely with Cyro Services, and they offer a program called SME Connect. And that actually offers some funding. So it's called Kickstart. And so people can apply for 50,000 Australian dollars. They need to provide matching cash, but that will give people $100,000 worth of work within the biofoundry, and then gives them hopefully a taste for what can actually happen. And they'll get super excited and they want to pay and come back. And there's other funding modalities along that way. The other major initiative for Australia, which was in the previous government, they're very interested in getting a lot of the research that's done in Australian universities to be commercialised. And so they put $250 million and they've funded a program of $250 million. It's called Trailblazer. And so there's 4 universities that are funded. We are part. They need to access Cyro facilities, each of these different programs. But there is one program run out of the University of Queensland, accelerating growth in Australia's food and beverage space, manufacturing. And so we'll be working very closely with that team. And that team involves the university, but equally startup companies. And so the biofoundry will get $3 million for new CapEx, which will be able to increase automation. And we'll also be giving, they'll also gain the companies the people who are involved will get about $750,000 in access vouchers to access the services of the biofoundry. And this is going to actually allow us to build a platform that meets their sort of needs. So that program's, they're still trying to sign the head agreement, but it is happening. Lessons learned. I think the contracting is challenging. We have to run on a full cost recovery model. It causes challenges for us for competitiveness. We're quite small and we don't have subsidies. So that makes it, people, and I guess the time frame, because we're small, and we don't have subsidies, we can't go, we're just going to give you for free, or very low cost. And so we do lose some business that way, but others see value in the services that we offer, move forward. Some of the challenges that we've experienced in contract negotiation, and sometimes the contracts are executed, but people want their work done, but they don't provide the material. And then, so things sit around for 6 months and then people complain about timelines. And so Janet has changed the contracts to say that the work will start once the material is provided, and it'll be scheduled then. But if it's not, it'll be scheduled when it suits, when we're able to take it on. And I guess our capacity to do stuff is limited by team size and the space that we have available at the moment. But this is all, we're on a path to growth and a learning opportunity. And that's about it. I'm happy to take questions and continue the conversation. Thank you.
Okay, thank you, Allison. All right, so you have a question? Somebody want to ask a specific question now? No? Okay, well, we can come back. All right. All right, we have two more speakers, and this will be a little bit of a tag team because one of our speakers is online and we have his slides. That's Yinji is online. But before I ask Mark to come up, let me just give a couple of introductions to the next two speakers. So Mark is a winner of multiple prestigious early career awards, Mark Blenner, that is, and his research takes place at the University of Delaware and focuses on synthetic biology and metabolic engineering of non-conventional yeast and bacteria and microbial communities. He has partnered at least once with Yinji Tang and in fact is working kind of hand in hand with Yinji on an Agile Biofoundry project. And Yinji is a professor at the McKelvey School of Engineering at Washington University in St. Louis. And Yinji's research focuses primarily on Euroia yeast engineering for synthesis of biopolymers and natural products. Also, he works in cyanobacteria for CO2 capture and biomanufacturing and the development of machine learning models for strain engineering and fermentation optimization. So I would like to have Mark come up. And so what Mark will do is control, at the end of his talk, he will control the slides that Yinji has provided, and then we'll have Yinji speak to those slides from the cyberspace. So, okay, without further ado.
All right, thanks.
Get your slides up here. Okay.
Great. Thanks, James, for the invitation to talk about this Agile Biofoundry project that Yinji and I have, and thank all of you for listening today. So I'm going to give very little tiny bit of the data on this presentation on our project and really focus a bit more on kind of the thought process as a user of an academic user of a biofoundry. And so in seeing the opportunity to try and develop a project that we could work with the Agile Biofoundry on, I sort of started by thinking about what their mission was and to think more about where we fit into that. And so this is just cut and paste from the Agile Biofoundry website, but I highlighted a few things that caught my eye as I read through this. And so you see terms like accelerating biomanufacturing, collaborating with industry and academia to rapidly innovate the development of bio-based products, and basically trying to help us go where we want to go faster. And so, reading between the lines, this really did seem, from my perspective, to be the kind of thing that was aimed at industry, right? This is aimed at trying to accelerate commercialization. And so where's the role for the academic in all of this? And so I think that from my perspective, and I probably shouldn't really speak for Yinji, but I think from his perspective too, we see that where we fit in is in really two different areas. That academics can contribute to trying to solve, I think, industry-wide problems in a pretty competitive way. And then the second place is in trying to help develop new technologies to accelerate what biofoundries across the globe are doing. And hopefully we get science papers out of that, and biofoundries get new technology that they can use to help commercialize things faster. And so the particular project that we proposed was one that Yinji and I had talked about for a little while, and is really kind of a case study of a failure. And the work started with this company Yinji had been working with called Arch Initech. They were trying to commercialize a process to make various types of carotenoids using Uroia lipolytica. Uroia lipolytica, for those of you unfamiliar with the organism, is a non-conventional yeast. It's probably the model, non-model yeast, I would call it. It's got a decent amount of tools available, and its main characteristic is that it's really good at accumulating fatty acids and lipids. And so hydrophobic compounds like carotenoids are logical targets for production in a host like this. And so anyway, so the story is that the strain that was developed made beta carotene and beta ionone, and it did so really well in lab scale bench-based bioreactors, getting things like 50 mg per liter beta carotene in a shake flask and upwards of 4 grams per liter in a sort of 2-liter bioreactor in a lab. And as this process was taken out of the lab and gone into larger scales, as the scale-up process began, the strain would consistently lose its productivity. Right, so there's some sort of a stability issue here. And since then, this issue has caused the company to fold up, so it no longer exists. And so we were curious about some of the underlying mechanisms for this particular strain and how it was unstable, and maybe what that would mean from a broader perspective about strain stability. I think Uroia lipolytica strain instability is kind of it's an unspoken secret amongst Uroia lipolytica users. And I think often we, especially on the academic side, we stop exploring once we get to a certain scale. And so a lot of these issues don't come to light. And so we're hoping to illuminate that a little bit with our study. So we wanted to answer some of these questions about, is the productivity loss genetically based or epigenetically based during the extended growth period? Whatever the changes are, whether they're genetic changes or epigenetic changes, do we see that those changes are uniform across the genome, or are they concentrated in certain regions of the genome? And how do those changes correlate with metabolic stress? We had some idea from the start of the project, and further support for this idea now, that various types of metabolic stress in various culture conditions can accelerate the rate of instability and the loss of productivity. So the team that we put together, the Agile BioFoundry has, I think James showed the slide of, I forget how many exact labs, but many other national labs are involved. And so in partnering with the ABF, we get to identify the labs that can help us out most in the most efficient way, because there's limited resources to do these projects. And so you can see kind of the workflow here, or the materials and human flow between all the different sites. And so Yinji had sort of the base strain. He sent it to me at the University of Delaware, where we've done some of the genomics and the transcriptomics. And then we've sent that on to the ABPDU, which is where the fermentation work is being done. Yinji sent one of his graduate students there for 6 months to learn fermentation. I'll say this is probably one of the biggest advantages from my perspective for a US academic lab, is that there's not really a lot of academic expertise in fermentation. And so partnering with ABPDU gives us access to that expertise that we'd otherwise lack. Alyssa is generating samples there, and then they're going to be sent back to Washington University to do some of the metabolomics, as well as to the Pacific Northwest National Lab. It's also doing some proteomics. We're going to do transcriptomics on these samples. And so I think this is sort of, when we talk about a distributed network of labs that form an integrated biofoundry, I think this is a really great example of that. The things that we're interested in looking at throughout our project, in terms of some of the simpler to control metabolic stresses, we're looking at the dilution rate, the oxygenation levels, the media composition, the pH, and sort of the analyses right in the bottom that I just mentioned already. Just a little bit of our data. We're able to take the really what was derived from a single colony and has been glycerol stocked and kind of passed around for a little while now. So we think it's aged a bit already at this point from the original work. But we can see when we pull out 3 different colonies from our stock, we can see very consistent decline in the beta carotene production, both at absolute and a specific basis. And this is highly repeatable under a controlled set of growth conditions. And so these are just simple like shake tube experiments that we did in our lab. And you can visualize the differences here. I think we were passaging these every day. And so they grow to mid log phase, then we dilute them back down and keep doing this for several days. And so by day 4 already, you can see the loss of the orange color associated with beta carotene. We measured using qPCR and gene-specific primers. We could see, particularly in the CAR-RP gene, the decline in the mRNA level. The genomics we have, we're still sorting through some of the data to get a full picture on the story here, but we see copy number variations throughout. Sometimes we actually see what looks like maybe an increase in the copy number, but eventually we see by this midpoint here, this associated with the loss of the beginning of the loss of the productivity of beta carotene, we see decreases in gene copy number. So this is we have just a couple of slides from Alyssa's work at the ABPDU. So she's there working on, I guess, 2 different types of reactor systems. She's developing chemostats so that we can control the growth rate in steady state. And she's using the Amber 250s for, I forget the way she described it, but for sort of semi-continuous fermentation. This was an interesting experiment. Again, we're just starting to, I think, scratch the surface of what some of this data means, but here we actually saw at some point there was a spike up and an increase in the productivity. And so we have these samples collected and are very interested in understanding metabolically what happened there. We see there was a small but really fairly insignificant increase in the cell density and a major drop in the glucose concentrations. And so the cells went into some sort of a mode where they really accelerated the production of the beta carotene, and then shortly thereafter have gone into declined past their initial titers to basically nothing again. So these are interesting things that we can capture by using these types of chemostat systems. The Amber 250s just give us a way of doing really nice controlled bioreactor experiments at higher throughput. And so this was just looking at 2 different media compositions and 2 different dilution rates a little bit. We'll call them dilution rates, but they're, these aren't actually operated in steady state. So, and you can see, interestingly, on the bottom here with a richer media and a slower dilution rate. Again, we haven't normalized this for generation yet, so but we're seeing, we're not seeing the same loss in productivity as early on. So again, this is suggesting the importance of some of the culture conditions for the stability of these strains. Okay. And then that's all I have for the data. And then just a few thoughts on our experience partnering with the ABF. And of course, we're happy to share more of this. I think we put an application in April of 2020 to, I believe it was the open call they have that is usually targeted and I think is better suited for industrial partners. We received notification just 3 months later, which is an incredibly fast turnaround time. So that was a really positive experience. The contracting agreement through the CRADA here, that took longer than I would have liked, but I don't know if honestly, I don't remember if that had to do with me moving institutions in the middle of all this, or if this was just the nature of how long this took to do. But this is probably similar to other CRADAs I've had established with the Department of Energy in terms of the time length there. So it's a long horizon just to get agreements in place, even with academic partners. And so we're sort of, I don't know, we're sort of maybe 9 months into the 2-year project at this point. Benefits that we see as academic users, of course, it's having additional access to expertise and equipment that we either don't have or have trouble accessing, or the funding opportunity requires us to provide a lot of cost share, but we don't have to provide dollars to do these experiments, and so that part's a big benefit to us. The data sharing, I think, between the labs and within the individual academic labs involved is very simple, and so that's potentially could be more difficult from a commercial perspective, but from the academic perspective, that's been a real benefit of the partnership we've had. And then the workforce development opportunity, I think it's very clear that much of our industry is moving towards a model where the strain development will be done in biofoundries. Biofoundry thinking and design of experiments is much different than kind of standard lower throughput strain developments in labs, and from my perspective. And so we need our students to be thinking at a different scale. And so they need the exposure to working with biofoundries. Also, there's the really direct opportunity for sending students, like, for example, to the ABPDU to learn fermentation in that particular case. The drawbacks we've had are, money, that we don't, from this particular opportunity, we didn't have any direct funding. So we're providing cost share and having to, pay our students from all sorts of different places to cover their bills. But I think a positive aspect on that drawback is the current NSF funding opportunity that's now available that Yunji's actually been able to avail himself of. So, all right, I am now going to turn over the audio, I guess, to Yunji, and I'll control his slides, and maybe just I'll do that so all of the animations are up.
Thank you, Mark. Hey everyone, good morning, and thank you for inviting me for this panel discussion. So now I want to spend a few minutes to introduce my recent project with ABF. This project is try to use machine learning to predict the engineered strain performance. And so as we know that when you want to scale up bioprocess and the engineered strain, actually the production has been controlled by many influential factors that including the react condition, stress condition, product toxicity, growth medium, metabolic burdens, and also the enzyme functions, and how you modify the pathways, whether you have what type of the synthetic biology component you use, such as a promoter or sensor, and also the genetic stability also affect the production, and also the what type of the substrate that you're using. So it is very difficult to use a mechanistic based model to predict all these influential factors that affect the cell production performance, how they affect the yield, rate, and the titer. So in order to solve this problem, and so we try to use machine learning based algorithm to enhance the model prediction and biomanufacturing. Next slide, please. And so the idea is like this. So we have the design, build, test, learn cycle for strain development. So we needed to have a good model to design the pathway. And so typically computational strain design is very powerful method. So you can build a genome scale model, and you can use bi-level optimization to predict all kinds of the mutants. And you can use the model such as OptForce or OptNock, and that can give you a lot of candidates, genetic candidates that may lead to improve the production. However, because genome scale model is not like the precise, we know that because the model is basically based on the mass and the energy balance. So, but there's a lot of other influential factors make the cell metabolism to be suboptimal. So in order to improve this genome scale model accuracy, we actually can use machine learning. So basically, the first step is that we will do the knowledge engineering. So we will collect all this data, and so the ABF actually have a lot of lab have this yeast manufacturing data. So we can get this data, and this data include all kind of growth conditions, bioreactor operation conditions, and also how they engineer the strain. So then we will build a database. This database actually we use numerical or categorical variables to reorganize this information. So basically, we will identify some features that can help the machine learning model to predict the cell performance. So we hope this project actually can be used. This model platform can be used to improve non-model-based production. So, for example, we want to engineer the natural product synthesis, and we want to engineer biofuel production. Whether using this machine learning method, we can actually increase the speed of the design, build, test, run cycle, and reduce this failed trials, and actually can help the strain to be more easily to be scaled up. For this project, I want to also mention that one of the co-PI of this project is from Lincoln University. So it is historically Black college and the universities. So Lincoln University is an HBCU school. So we actually will offer the opportunity that Lincoln University students, most are minority students, will work in ABF labs and for the summer research. So these undergraduate kids can actually get a very good training in the biomanufacturing, and so make their career to be more promising. And so I'm very excited for this opportunity. That is all my presentation. Thank you.
Actually running a little bit ahead of schedule. So if there are any technical questions that we'd like to entertain at this time, maybe we can get some for the speakers. It looks like we have a taker.
Thanks so much, Mark. Yeah, sorry, pack up.
There we go.
It's a very cool project, and it's clear that the availability of access to both the Agile Biofoundry and the production unit, whatever that is, ABDPU or ADBPU.
I always have to look up the acronym.
I never can do that. Yeah. What would you have done instead? Absent the availability of the national lab capabilities to you guys, and I understand it was cost share, or it was, you know, NSF had to come through to actually pay for the research. It wasn't a funded research program, but absent the availability of that platform, would the work have just not happened, or was there other?
Yeah, I mean, I think the work in its current form probably wouldn't have happened. I've pitched a project to the NSF opportunity that wasn't funded. That was there was a lot of build stuff in here I'd like to test out that we didn't get to do with the funds that were available through the original ABF opportunity. But yeah, no, I think Yinji and I both probably wouldn't have pursued this just yet. Yeah, we would have looked for a different opportunity. So it's, you know, the availability of the resources definitely enabled the project.
Great.
Any one or two more questions, and then we can break for coffee and come back to the panel discussion. No? Okay. So this gives us a little bit more time to flourish on the coffee. And so maybe this is a nice way of kind of the other side of timing, where yesterday we were perennially running late. So I'll let you go for the moment, and then we'll come back at 10:40. Is that the time? 10:40 to kick off the panel discussion. Okay, thanks. All right, so while we're waiting for everybody to come in, I would very much encourage you to, if you have a question at the ready, if something is on your mind and you are interested in asking our esteemed panelists your question, please make your way to the microphone. We can't pass the microphone around because the camera is stationary and it's on the mic. So if you do have a question, please do queue up and get ready to offer your question. So today's panel is a decidedly different format than the roundtable kind of brainstorming that we did yesterday, but nonetheless, I very much am hoping that the questions will generate more questions and even a few answers here and there, and give us lots of food for thought for how to leverage the biofoundries throughout the world to really respond to the mission of sort of the broad mission of biofoundries to enable the bioeconomy, to lead us to different routes and improved routes to sustainability, alternatives to fossil feedstocks, et cetera. So there is lots and lots of work to be done, and it's so great to see some of the biofoundries that are represented by way of the panel, and some of the biofoundry users that are represented by way of the panel, to really state their case and to provide the kinds of insights that only they can offer. So I see that we do have a couple questions at the ready, which is fantastic. Again, let me introduce the panelists. We have on screen Yinji Tang. Thank you, Yinji, for joining us.
Thank you.
And alongside Yinji, we have Mark Blenner. We have Allison Rice, and of course, Marco Storch. So without further ado, looks like we have a question. Please, when you step to the microphone, state your name so that folks online can know who the question is coming from.
Hi, I'm Kellen from DTU in Copenhagen. I'd like two questions. One direct question to Marco about the you said you talked about people getting seconded into the biofoundry. Like, how does that work? Like, in a financial sense, do they pay to just use the equipment, or do they just pay like consumables? How does that kind of work, that sort of deal? And then discussion about like a few of you raised about working with businesses like SMEs and stuff. Again, I guess because about financials and stuff. Do you see then like that your biofoundry is moving towards to just being like a profit-driven biofoundry to work with just businesses? And like how much of your biofoundry operations are working with like businesses? And is that the future, do you think, of the biofoundries that you're all setting up or have set up? Cool, thank you.
Cool. Thanks for the question. Two questions actually. Yes, for the secondment, right? So that's for us kind of a flexible way of kind of enabling young companies, right? So we try to kind of, keep this as open as possible, as friendly in principle. Often it's part of a bigger project, right? So you describe a bigger project that you try to deliver. Part of that will be a secondment, right? And what it comes with often is like a package around like access and so on to equipment, consultancy and management support for the projects and so on and so forth. So it's often kind of rolled into like a bigger project description, in fact. And then to the second question, maybe certainly not just profit-driven, right? So I feel like a lot of biofoundries think hard about sustainability, right? So that's like one of the major missions. Like, I think a lot of places got like a good starting point. Sometimes there are really generous government support available to like get going and build infrastructure and so on. But it costs a lot of money to keep it going, and also like to kind of fulfill what the promise actually is here, right? And so I think I take comfort in the fact that, in my mind, the startup community will be a major vehicle to deliver the impact that we actually want to see, right? Like kind of growing the bioeconomy and kind of, reducing the carbon footprint and so on and so forth. And so in that sense, I think it's actually spot on in terms of the mission, right? I think there's certain things where these startups are just much better than biofoundries would be, for instance, or other organizations. And so it's really kind of this kind of nice synergy in between, on the one hand side, having like a welcoming environment where you can support these activities, provide training, also learn yourself about the domains that these startups really specialize in. So I think that's hugely beneficial for us as well. And I guess there's just a financial element to the transaction that ultimately supports all the other good work we do that is more on the educational side and the academic side.
Excellent. Okay, thank you. Next.
I'm Ben from Concordia. I've got like 20 questions. If no one else comes up, I'm just going to keep talking. I'm curious if you engage much with, and this is a question for all of you, if you engage much with VC firms, and if you do, what that looks like, and if you don't, what you wish it looked like. Thanks.
So Cyro has a partner, Main Sequence Ventures, and so we work quite closely with them. Engagement with them looks like talking to them. I guess one of the key things, they've been a major player and a major supporter for biofoundry and synthetic biology in Australia. And so Phil Maul, who's one of the partners, he made the point that in 2016, there was no SynBio startups in Australia, and after the 6 years of the SynBio FSP, there were 10, and there's a lot more VC capital coming into Australia and a lot of excitement. So I get to see Phil fairly regularly. We talked about the idea about pitching to the UQ MBA. He was part of that sort of idea. We're talking with them about the bioincubator. So I guess it's an ongoing conversation. That, and not something you pick up, once a year. It's once a month sort of conversation, and it needs to be an ongoing conversation where you build trust, and they see what you can do, they know what you can do, and they're really strong advocates for you.
Ben?
So yeah, I'm Lachlan from DTU in Copenhagen as well. So this is a question for kind of everyone on working with business. Something that's been like a discussion point or a constant thing we've had to think about is confidentiality and IP, and who has access to places that have very valuable strains that companies are quite concerned about. It's been more of an issue with the larger companies. So just wondering if you have any comment on how that's been, bringing lots of different people in with valuable property and IP and strains that someone can take a tiny amount of and then grow as much as they need.
Well, I guess I'm sad to report that we don't have the $1 million strain in the freezer just yet. So it's not a direct concern at the moment, but I think it points to a really important question though, right? So I think a lot of things here in terms of working with also larger companies on these scale projects, right? There's certainly a professionalism required, where it's not obvious that a biofoundry that's growing out of an academic environment would have everything in place in terms of like the legal support and the policies and the QCs and what have you, right? So my feeling is, and I guess like in our specific case, like that's the advantage of startups, right? So they are as flexible and on the learning curve as we are ourselves, right? So that's a good team up there. I can well imagine that like for like, if GSK comes along and they go like, I think you could do cool stuff, but we just don't want to take the risk. That might be perfectly possible. And I guess it's like one of these things where there's so many activities that we could invest into and getting ready to support the projects properly. There's really just a question of the timing and like whether you want to spend your time and energy. And my feeling for the London biofoundry is probably that we're not quite there yet, where we want to prioritize that. But I think it's very important to keep that in mind, that that could be the direction of travel, right? And ultimately you want to be able to kind of provide the same standards also to younger companies and so on, right? So it would be nice if from these kind of activities, there's a learning experiments that also trickles back into even the academic environment, like to kind of get to these standards, right?
We don't have the $1 million strain yet either, but we're aiming for it. I think that the challenge for us is we're quite small. And so with a team of 5 actively working in the biofoundry, to bring in, to have like somebody like seconded in is quite challenging. And we've thought about that, because how do you, partition and keep confidentiality in a very small environment where you have to share. People need to know what you're doing. I guess the other thing I'd comment, we are set up to, Janet and her team have thought very long and hard about how they want to work. So they're set up with success in mind. And so keeping, really great records, the IP, the legal support, all that sort of stuff is front of mind to help when that, the $1 million strain does come in. Do you have any comment?
I have a quick sort of follow-up to that. And for the so-called non-practicing entities like the national labs and universities, the ones where IP might be generated, if we're doing it right, but will ultimately license that IP out. I'm curious to know from Yinji and from you, Mark, what kinds of conversations take place on that side of the equation when it comes to understanding how to enter into these agreements and the intellectual property kind of implications.
I think every university is probably a little different in how they expect their faculty to evaluate these sorts of things. In our particular case, we didn't have any background IP really that was, I think, worth protecting at this point. So it's a matter of, looking at the ownership of the generated IP and whether or not we, so my tech transfer office relies a lot on the faculty to determine the real potential value that could be generated. And usually just goes along with that assessment.
Okay.
I don't know if that makes us easy targets for the rest of you in this room. So that's really the extent of the conversation. I'd say we're kind of, at least from the multiple stops I've made now, pretty underprepared for having real serious thoughts and policies around protecting the potential IP that comes from something. I think when there's IP there already, the universities get a little more careful about what goes out.
So if it should come up that something really interesting arises, then everybody's hair is on fire to try to figure out how to value it or how to license it.
Yeah, pretty much.
Okay. Understood.
I don't know if Yinji has a different experience with his institution, but
I agree with Mark's comments.
Okay, thank you, Yinji. All right, Vincent.
So I have sometimes I feel the foundries and the business models that were described, or the funding model that was described, sometimes are at odds in the way they operate. And let me explain that a little bit. We heard through some of the foundries, and even from Ginkgo yesterday, we have, in order to be sustainable, we have to have these 100 projects or these 20 companies in emails. And here's an example. We have 2 examples here of academic investigators coming up to a foundry and saying, I need this project done. So the power of the value of a foundry is doing the same thing over and over and over again and having things that are standardized so you can accelerate and scale. If you have 20 people knocking on your door all the time and they're all asking for something slightly different, and you constantly have to develop or adapt your workflow, you lose that value of a foundry. And that every new project is a new development, is a new, and it's hard to scale that. So I'd like to hear a little bit about, from Mark and Yinji. So the process at the Agile BioFoundry is you wrote a grant and you submitted it, and Agile looked at the grant and said, a panel looked at the grant and said, do we or not fund this? It's an interesting project. But then probably what I suspect what probably happened is you guys showed up with a round peg and they had a square hole, and you had to, I expect, I'm not sure, maybe you can correct me, and if that's true or not, and if it's true, how much of the shaving of your square peg or shape did that take in order to onboard your project? And if this was big, and you can assume that every project would be like that, then again, you lose that value of a foundry. So I don't know if that's a question or comment, but I want to hear more about, first of all, from the business people that try to bring all these projects in, and second of all, the people that are trying to bring projects in, to see how that fit from the hole to the peg is complicates things.
Yinji, would you like to kick us off?
Yeah, I think so. So this is what I'm thinking. So because we're in the university, so we're more interested in fundamental research and try to understand some kind of the rules of the life. And so we're not like an industry, so we want to actually make the profits, but our knowledge, our modeling tools can be useful. So I think my thoughts is that our work is complementary to say the industry, and we can provide very useful knowledge and information. And so but we're more like interested in the publication rather than some like IP or stuff. So this is what I thought. Maybe Mark have different opinion.
Yeah, I mean, I think we don't have a bottom line the way a company is going to, right? We don't have investors really. I mean, we kind of do, but there's a lot more security in what we're doing. And so I think one answer here is that we, you know, our round peg, we can mold to something different, right? We want the information, and I think it was beneficial for us actually to be able to discuss with the various people at the ABF ahead of time what our idea was, and we actually, our proposal is crafted towards what the capabilities are to some degree. So yeah, I think we probably did do some shaping and the resources. Like I think I've said a couple of times now, I really wanted to build a bunch of strains to further test some of our ideas, but it wasn't fitting in the budget. And so that was something I had to jettison and wait for another day. So I think that mismatch is easier to reconcile when you're dealing with an academic partnership with a biofoundry versus a company.
Maybe James, you can follow some. There's nobody behind me, so I can keep one. Maybe you can follow up on this. So I was privy a little bit of the process that you guys do at Agile, and one of the things that I noticed or I realized is that the projects that get submitted to you, the grants from either industry or academia, propose a certain technology development or use of a tool or something like that very often. Does the foundry pick and choose the partners they want to bring into their foundry based on the new technology they might bring in? Or they pick and choose companies that come in that can use the existing technology that you have? So are you using projects and partners to drive forward the value of your foundry or not?
Right. I'll answer that. I did want to kind of just respond to your square peg round hole question, which is really an important aspect of how we scale this and where it's not scalable, because part of the secret sauce of these sorts of relationships is the relationship building, and you can't do that in a McDonald's style fashion. That has to happen kind of one at a time. And I think that that's something that Yinji and Mark are alluding to, is that we had conversations amongst experts as to how best to craft a proposal that responds to the capabilities of the ABF and puts their best foot forward so that they can be competitive in and amongst an oversubscribed set of applications. And that exact same thing happened for each and every application that we received. And it is not a light lift. And so to your point, that costs money to do that, often sort of absorbed in a way that is difficult to track, but arguably painful.
I think this speaks to the agile word in the agile biofoundry. There's a desire, I think, that I've noticed from the ABF scientists to expand their skills, their tools, and so to me it's a little different than I think some of the other biofoundries we've heard about that have a much more of a business driven kind of concept.
Right. Yeah. Addressing the high risk projects and understanding how to avail the capabilities of which there are many at the ABF on behalf of a wide variety of projects. So yeah, that sort of is a square peg round hole type of question. And the second question you were asking, Vincent, was, can you repeat that? The one?
So whether or not you pick and choose projects coming into the foundry as a means of developing new capacities, new workflows, new
Right.
new
Well,
technologies.
the new capacity question is definitely a very valid one, and one that we, that the selection committee does take into consideration. But that's only after we have had our external review panel evaluate and really score and say, these are the ones that are ranking the best. And we step through that very, very carefully and systematically to understand amongst that top group, which ones might we really want to champion because it helps us balance resources. It brings new capabilities and it helps us onboard a host that is very attractive, sort of broadly speaking. So it's a calculus, and there's a lot of decision making that takes place in order to get that right portfolio. Yeah.
I'll also point out, having served on one of the review panels in addition to now having a project, the external panel review definitely has people with different mindsets of what's important in those proposals. And I think those, James and the rest of his team take that feedback and try and make the most of it. So like my mindset is more of like I think that there needs to not just be a commercial potential. I think there needs to be a commercial potential with a benefit to the ABF. And that's not a commonly held viewpoint on the review panel I was on, at least. I think we had people from industry and they had much more of a, no, what's the bottom line here? And I'm like, well, the bottom line's okay. You got to have something that's there, but I also want to see growth in what the capabilities of the ABF are.
Right. Yeah. So we don't want to sort of eat our young, as they say, and it's important to have that long game consideration. But it's also important to acknowledge the fact that if we're going to do this right, commercialization is a thing, making money is a thing, and so we have to balance the long game with the shorter term kinds of goals and objectives that the market demands. So.
Oh, maybe one last, because then I'm going to sit down and you're not going to hear from me again. I'd like to hear from Mark and Allison about kind of the similar question. So if you look at a biofoundry workflow, it's endless, right? There's so many tools and methods and approaches that you can bring into a biofoundry. So how do you approach that? Is it project/client driven, or do you take a chunk of your budget, put that aside and say, I have one or two people in the foundry that just develop workflows and methods and everything else that can be used afterwards to attract new clients and new contracts in the foundry. That's my last question.
So I think we're still in the sort of the growth phase, and I would say that from a BD perspective, each project is considered, and this is a great idea. Let's bring the technical people in straight away, so that we can actually work out whether this is something that's going to fit. I would also say that we have an overarching impact pathway, which guides what we want to do and where we want to go. And we are responsible. We have to report back to our funding, overbearers about what we've done and where we're going. And so how does this fit with the CSIRO mission? Does this bring benefit to CSIRO? Does, all these sort of things, the questions comments you were raising fit into the discussion. But technical expertise comes in very, very early. Is it feasible? Can we do it? What is this something we want to do? And can we benefit from it?
Yeah, I think a really fundamental question, right? So I guess it might really depend, like, again, on the type of foundry, the type of team, and the ecosystem, right? So that will determine like what you want to emphasize. I think in our case, I guess like most people have like an instinct where things might go, right? So that kind of guides you a little bit. I think there are, when we think about like where do we want to invest resources into, I think we're always looking for like multiple outputs of that. We always want the synergy. We don't want to just do this single purpose thing. I mean, we might do if there's a very specific academic project or so, but I think that's something that kind of safeguards a little bit against that. I think like one example, for instance, like Keltum from our team, she also presents a poster here on kind of, high throughput mini prep. Like that's something that in our view is still not completely solved, right? It's something that's really essential to a lot of different workflows. So like picking out these pieces where we can see how different upstream and downstream workflows might benefit. So I think that's probably like the easy ones to pick first. And so definitely that's what we also do emphasize most. And in terms of which kind of workflows the customer decides. So if we decide which customer to take on, I think that really links back to this cultural question, right? Like I think within biofoundries, maybe there's already a form of consensus around, like, let's say, Golden Gate type standards and so on, right? And once the critical mass is there, then there's actually no reason to move somewhere else, right? And you just like lock this in. But I also think that the most interesting projects really are the ones where you take like your bread and butter applications, right? You build your plasmids, but then like the interesting bit happens after, maybe in the assay development and so on, right? And I think kind of focusing on these type of projects that allow you to always push boundaries a little bit has a lot of useful functions, right? It's also, I'm always thinking about like our team and making sure like that it's worthwhile the time, that they also like learn something on the way. So I think there's a lot of these considerations, again, coming back from the ecosystem, from the context, that help in our case inform this.
Thank you, Marco.
Okay, I got one more question. This is a question for Yinji and Mark. I was curious about how working with a biofoundry has changed your approach to thinking about your research and thinking about onboarding a project. You can speak to that at all.
Positive, hoping Yinji will jump in, but. I mean, so in my mind, I think, you know, it's definitely made me more aware of overall foundry capabilities. Like, I think I have a very general sense, but I have a more specific sense of what foundries can offer now that we've worked with. We've spent time looking at what ABF does. We've thought more about, you know, read some of the papers on how people start up their foundries. I think Claudia wrote one, was good. So, you know, it hasn't made a huge dent yet in my portfolio of research because it's not a capability we have a lot of access to still. I still have to, if I want to access ABF, I still need to write another winning proposal, or and even, you know, I tried writing one, didn't. Like, Genji was successful with that exercise. So it's starting to shift the way I'm thinking, but hasn't really made a huge impact to me yet. But now that I can see what's possible, like I decided to come here and I agreed to speak because I wanted to learn what it would take to start some form of a foundry in Delaware to catalyze an ecosystem of SynBio around us, where I think there's already elements in place. So it's a slow movement, but I think it is shifting how I view what's important in research, what kinds of problems we can tackle, the way we tackle them. So yeah. Yinji, do you have any thoughts?
Yeah, so from my experience is that before, when I started my career, my most of work is on modeling, metabolic modeling and metabolic analysis. So my work is very, very far away from industry. But since like 2 years ago or 3 years ago, I started to work with Mark. And actually, my work is more and more close, like try to solve some industry problem. And this is the ABF actually provide a very nice bridge to link this academic work to the industry application. Secondly, I think that because in the academic field, especially like my lab, we don't have a lot of funding, so we cannot do very advanced research. We have some idea, but we don't have the equipment. We don't have the resource, say, to do some, let's say, omic study or some kind of advanced modeling, and we don't have enough data, say, to train the model. So with the ABF, actually, all these kind of issues or problems have been well resolved. And also, I'm very lucky to get a grant from ABF. NSF, this grant is on the machine learning. This is a completely new field for me, and this is new things, actually very new things. I'm very surprised I got this grant. And so I think the ABF for this type of the work is actually also helping me to open some new frontier and a new area and to expand my career and my research funding.
Thank you, Yinji.
All right, next question.
Hi, I'm James from the Concordia Foundry. I just had a question whether, like, what elements of the sort of surrounding startup ecosystem you felt help or hinder your ability to partner with institutions? Because I imagine that people in your area are the ones who are most likely to partner with you. So what exists around you for supporting startups, I imagine, is significant.
Yeah, so I guess like we're really quite fortunate in the sense that we are located in the innovation hub, right? So that's really a building where there's like 10 of the startups we're working with on the same floor or the next floor, right? So it's really this proximity that's quite essential to make things easy and practical. So I think just the space, having that there for people to have access to, and also maybe at rent rates that make sense for a startup scene, right? So I think that's one thing. I guess like then you need a source of startup creation, right? So that means, you know, universities that create like the talent pipeline, the interest, the urgency. And then I think also, I mean, I guess we touched on it a little bit, the kind of funding opportunities, right? So I guess within UK, there's a program called Innovate UK. So it's quite a sensible way of trying to get some non-dilutive funding. So in the range from 200 to 300K maybe, that a lot of the young companies working with are quite successful regularly, actually, it turns out. And so I think certainly the funding time and kind of the runway are important, but also just like maximum flexibility, right? Because like those young companies move all the time in the first 1 or 2 years. And that's also very entertaining, I guess, to some extent, right? But it just means like the program you try to shape around it to support them somehow also has to reflect it. But that's what I would think, like, so the talent pipeline, right? Space is quite essential in places like London for sure. And then of course, like some form of funding could be government, non-dilutive, or a VC.
So for us, we were lucky. Paul Birch was a science director within one of the business units within CSIRO, and he saw the potential of synthetic biology about 6, 7, 8 years ago, and then he became chief scientist for Queensland, and he spoke very charismatically to the Queensland government and got them all super excited about the potential of synthetic biology, and particularly for its the opportunities that it creates to build new jobs and growth in regional areas. And so that became, that got the attention of Queensland government. And so I sit on an interdepartmental government working group that meets monthly, and we are committed to try and build this bioincubator. But at the same time, Trade Investment Queensland has appointed a commissioner in North America whose job is to drive synbio investment into Australia. And so all these things are happening in parallel. And we have our university partners, there's University of Queensland, Queensland University of Technology in close proximity. We've also got a major health research institute, the Translational Research Institute, and they've just got funding for manufacturing so this is where Gardasil comes from. And so all these sort of partners are happening and talking at the same time. There's a probably fairly strong IT startup ecosystem in Australia, and particularly in Queensland. But I guess what we're seeing now is there's this real need for rentable PC2 space to bring in the startups, because they're actually going overseas. And I guess my big problem is that everybody's super excited, but nobody makes a decision. So I just really want somebody to make a decision, line in the sand, we're going to do something, and here's the money.
Excellent. Thank you. All right, Janet?
Yeah. So I'd like to echo the points about round peg, square hole, and the absorption of costs of onboarding new projects. It can be an interesting challenge to work out the balance of how much you charge your client for onboarding something and how much you absorb yourself. So I like that point. But in terms of, I'd like to address this maybe to Marco and to James in particular, because I know how we work. So, lost my question. Yeah, so Marco made the point about there was a contract you had up on the page where you had like the pilot followed by like the negotiation periods, et cetera, and then the larger body of work. And I think that kind of taps into what we're asking here about how much the partners, how much the biofoundries should mitigate the risks associated with your partners' projects. How do you go about that?
Yeah, so I guess like that's a really good question. And I think that's where I guess like running these type of activities in a biofoundry like turns you in a little bit of an entrepreneur yourself, right? Like you kind of in startup territory where you have to kind of make judgment calls on risk associated with those, right? The hope would be that like within your environment, both in the biofoundry and the academic support environment, there's kind of enough knowledge that you're in a good position to judge if a certain proposal might be doable, right? Plus the kind of experience you have running experience in the lab. So that's good. I think it comes certainly down to like the type of relationship, right? So if you would have a partner who is very hard-nosed, I think it would make it very difficult for us to come to an agreement that we feel comfortable with, right? But we've been quite lucky so far with having people that seem to appreciate what might be possible and therefore like also understand like our process in the sense that you kind of have a de-risking phase where you just have also a chance to establish the culture. What does it mean to have somebody in the biofoundry lab? How does the reporting work? Are things as speedy as biofoundries promise, right? And so on, like having a little bit of a test phase and just creating hard data that both parties can rely and build scenarios on, right? And so I think in that sense, like this one example where we had like a short period kind of proved it all out, and then having a project that's technically similar so that you feel like confident that you can be more ambitious and kind of promise that might work. Of course, things are always milestone, right? And so the agreement is that like if at some point the project moves too much or like other things change, right, there's always the chance to limit exposure for the client. But it's certainly one of the very exciting elements to the whole kind of environment. I feel it's really like where kind of, you know, have like a mini startup within your foundry for each project, right? Because you have to do all these things and hope to get it right.
You know, when it comes to kind of risk mitigation, because at least as of so far, the projects that we have taken on have gone through a competitive application process. A big part of that is having the applicant identify the risks in the projects and identify the risk mitigation strategies. And in fact, that portion of the application is weighted most heavily relative to the other scored portions of our applications. So it's something that we take into careful consideration, and the commentary that we receive back from experts on our external panels are incredibly helpful. So there is quite a bit of effort that goes in and thought that goes into understanding the risks associated with a project. We've been very lucky in that, by and large, the projects that we've received have been very well crafted and articulated around risk. But part of the selection process does go into what kinds of risks we're considering taking on as part of that project, and how that kind of go, no-go opportunity or series of strategies is sort of laid out in the statement of work. So, yeah. Okay, next question.
Hey, I'm Aksa from the University of Queensland. So I think through all this session, I think it's clear how biofoundries can accelerate this development of commercialization of some products for industry startups. And in terms of engagement, let's say in academics, what is your approach? What is your vision in terms of like, is this biofoundries really also helping academics to accelerate discovery and create like a new science and it's affordable for them? Or on the contrary, is actually creating a gap and actually getting a disadvantage to those labs to have the money to put that one compared to other ones. So how can we make this more affordable? And what's your vision on that? Or is not, or you think that is out of the scope of a biofoundry?
Mm-hmm. Yeah, I think that's really a very important question, right? Because I think biofoundries only exist because of the community, right? And so it's really important to keep that in mind going forward. I think we talked a lot about like the kind of commercial side of things today, right? And I think that's also interesting and important to work towards sustainability. But I mean, as I mentioned, like in my slides, like we engage a lot in grant writing, right? So, and that's a very open process, right? And so if you get a grant funded, we can do it, right? I think it comes a bit down also to policy of the university, right? So any university could go like, we think a biofoundry is so essential and so important, we just co-fund it and that's it, right? So the community have the chance to do that if they want to. I think that's tricky so far, I guess. But there's quite a nice example, like in Imperial College, I feel, where Professor Jeff Baldwin runs a student foundry. right? And so that's then something that also is located slightly differently in the area, so it's accessible from a different group of people. And so that has a much lower entry barrier potentially, right? And kind of gets people started. So I think there might be kind of some form of compartmentalization, just because also the user groups are different, right? So like if you really emphasize a lot like the startup side, that's like a very different working from like working with academics, of course, both in time scales and kind of the type of work and so on. But yeah, I think our approach is like to be as open as possible and support grants, and that's ultimately why we can't do it for free, because we're not co-funded. We're not a facility, so we can't kind of charge it back to grants. So that's like our approach, but I guess that's very specific to each university and environment.
Yeah, I think we have a similar sort of problem. And I think another way to tackle this is with the grants, is with the grant review panels. And so, you know, what funding in Australia is exceptionally competitive, and its success rates are down below 15%. But so if people put in, they need funding for a biofoundry, the people on the peer review panels, because particularly with the Australian Research Council, they don't, it's a broad, you know, sector specific funding. They don't know what a biofoundry is, and they don't know why they should pay a significant amount of money for you to do this, that you could, you know, have 10 PhD students do. And so it's a mindset change that needs to happen. And I think it's that education of, you know, the benefits of the biofoundry, how it's actually going to accelerate your academic work that might then have this sort of commercial potential later on. And it doesn't need to have that direct, but it's the real mindset change. And until peer review panels actually see and value that, and you actually put it into your grant, then it's going to be quite challenging. And as Marco said, you know, we're governed by business rules. You know, we have to cover our costs, and we need to be self-sustaining. So whilst we can do in-kind contributions for some stuff, we do need to be paid. And that's the challenge and the paradox that we work with.
Accessibility, yes, accessibility is an issue. I think I have a lot of thoughts on this, but you know, I think right now the costs are too high, and the scale needs to be too large to make the costs even come close to making sense for most academics to get involved in most biofoundry activities. As a result, we're not answering the kinds of questions that one could answer with the power of biofoundries. We're solving commercialization problems, which is a great thing to do, but we're missing out on really advancing science through the innovation that comes with the biofoundry system. So I would like to see people who are at this meeting and not at this meeting contributing to driving the costs down of using a biofoundry and to make that more accessible across the board. I hope that gets in the report.
So we are actually 11 minutes over time. So at least by my stare at the watch. So I think that we might have to cut it, but I'll leave that executive decision to Vincent.
Short question. You each get a magic wand, one wish. More biology. All this, excuse my language, all this fucking work that goes into building a biofoundry. Enormous work in building automation, enormous work in integrating instruments and capabilities. If you had one resource to apply, people, money, Super Bowl ads, what do you do with your magic wand? Each of you, quick, one sentence. Start with James.
Super Bowl ads. No, but seriously, outreach is a huge thing, and it's often undervalued because everybody's got their head down and working hard on, you know, that capability that they are masters of, what have you. But yeah, getting the word out in one form or another.
I already get the answer of driving costs down, period.
Yeah.
But I'll say on the analytical side, to have analytical methods, whether they're biosensors or some sort of a new advance in mass spectrometry or chromatography, that would really accelerate that part of the cycle.
I will say space to house the automated equipment with the people to do the cool science.
Is that all right, Janet?
When they're out of space in Australia, I get really worried.
I think on
Perfect, nothing rusts.
I think on this one, on the team really like kind of being able to underwrite excellent people for as much time as they need. Like that's a big thing. And that would really transform it as well in terms of.
So
longevity.
underwrite your users, not to give them more time. Sorry.
The people, like the team,
Support
the core, sorry,
on
the core team, yeah, supported better.
Okay. Any last question really quickly? Are we done? I know I have a ton of questions myself, and thank you so much for being here as part of this workshop, and let's give a nice round of applause to our. Thank you so much.